Okay, thanks a lot. Do you hear me? Yes. It's working. Okay. So what I will lead you through our attempts to understand the entire protein connectivity which underlies the process of cell death, what happens during pathological conditions, and can we plan some therapeutic vision for the future in order to use this information on programmed cell death. So here you can see three basic biological processes. Two of them lead to the demise of the cell, either by its fragmentation into small pieces, apoptosis, the caspases, or by rupture of the extracellular membrane and the release of the intracellular content outside. So those are two well-known death modules. There are more in the literature right now, but I will focus today on those two. And the third biological process is rather a survival process. It's an attempt of a cell to cope with the stress, different stress conditions. Here, the cell is producing small vesicles called autophagosome that engulf intracellular components, deliver them to the lysosome. It's one sort of recycling of intracellular components, getting rid of damaged components like mitochondria, and so on and so forth. So this balance, this interaction between the three forms of biological process determines whether a cell will live or die under certain stress conditions. Now, a bit about autophagy and how from there we proceeded towards the entire battery of cell death and survival in the system. So you can see here that what happens is double membrane vesicles, which appear usually from the ER. The membrane is growing. It's engulfing intracellular components into this structure that is called autophagosome. This autophagosome then fuses with the lysosome, forming this autolysosome, where the lysosome degrades all the contents. And this is one way to restore building blocks and energy, for instance, during amino acid starvation or other stress conditions. Now, this process is highly conserved in evolution, first identifying the yeast system and then in the mammalian system. It is a mechanism of lysosomal degradation, as you understand. It has a lot of implication in restoring cellular physiology and its inactivation in pathological states. And there are a few types of autophagy. I won't get into it, but just remember that there is selective autophagy, where certain organelles are now driven to the lysosome, and are also non-selective. And the non-selective, the entire bulk of cytosol and organelles is driven to the lysosome. Now, the work of Asumi that received Nobel Prize 2 years ago, in fact, shaped the current status and what we know today on autophagy. And they here discovered the basic machinery of the process, what we call the ATG genes, which drive the process and is responsible for that. So if we look what we know today on the ATG genes, we have two groups. We have the basic machinery of autophagy discovered by Asumi and colleagues, first in the yeast. Then the mammalian autologs have been discovered by many, many groups. And you see that here, those 31 ATG genes that drive the process. And there is this second layer of regulators of autophagy that couple the extracellular signals to the basic machinery. And we, for example, have identified a few of those regulators, and I will tell you about one example, how we did it and how we got to understand what couples in mammalian system the extracellular stress signal to the basic machinery of autophagy. Now we know that from many, many groups, we can align the different autophagic genes along the different process of the system. For instance, we know the induction nucleation process, which initiates, we know which genes are responsible for them. The membrane elongation and closure, we know which genes are responsible for that. Here you have the basic machinery, and you have the regulators in blue. So for instance, just mentioning a few complexes in the system. So the ULK1, a serine kinase that is very important in the nucleation process. It's at the beginning of the process. We have the Becklin complex, another complex that turns on a PI kinase, a lipid kinase, and is very, very important in the initiation of the process. And those ubiquitin-like machineries that participate in the elongation process. So we can assign different genes to their function in the process, and we have the regulators that regulate them and couple them to the extracellular signal. If you just look at that, you see that especially in the nucleation step, we are not dealing with linear pathways. We are dealing with a network of protein-to-protein interaction. A lot of interactions which result either in phosphorylation, in pink, or in ubiquitination. So we are dealing with a network of protein, and this is the theme of my lecture today. We are not dealing with linear pathways. We are dealing with complicated networks that we want to resolve, understand the structure of the, and then pinpoint the soft spots in each network. So we have the network that underlies autophagy. You saw the genes. Similarly, we have the networks that underlie apoptosis and necroptosis. Here, the caspase family. Here, the RIP kinase 1, RIP kinase 2. All of them are organized in complicated networks of protein. And a second theme that I want to introduce, there is a lot of crosstalk between those networks. They are not independent. They interact with each other. And some of the crosstalk, for instance, is by direct link between an autophagic gene and an apoptotic gene. For instance, we have shown several years ago that NTG12, one of the ATG genes that participate in the construction of the membrane, interacts directly with the BCL2 family of proteins. Another caspases can interact with the backlin. So there is a lot of direct protein-to-protein interaction linking all those pathways. And if we want to understand when a cell will die or live, we have to look at the entire repertoire of genes. And this is why several years ago, we constructed this map. You can see the autophagic genes that I've talked about. Here, the genes that drive programmed necrosis. One of those is the necroptosis. And here, the genes that drive the apoptotic process. This is a map that we have delineated based on hundreds of curated publications over the years, including our work as well. And as you can see, this is a protein-to-protein interaction map. Protein interacts with protein. Sometimes the interaction culminates in phosphorylation, sometimes in ubiquitination. Sometimes this interaction culminates in proteolytic cleavage. So this is an entire map that describes the status of this, what we call the program cell death map. And you can see already here that there is crosstalk between the models. Here, crosstalk here, a crosstalk here, a complicated network that delineates the decision between life and death. The network is obviously turned on by a stimulus. So it could be cytokines, it could be DNA damage, it could be a drug target in cancer, to which I will allude in the last part of my talk. So there is input signal that turn on the network. Obviously, not all the models are triggered in each cell, in each stimuli, and this is one of our purpose, to understand how the models are lightened with different inputs. And the output is controlling the percent of cells that are dying in the system. How many die, how many are viable, and so on and so forth. So one of our targets, or one of our aims, were to convert this static map. This is a static map taken from hundreds of publications, how to convert it into a dynamic map that measures in real time in cells the profile of protein-to-protein interaction in response to stimulus. Can we do that? Looking on those 100 genes that participate in apoptosis, necrosis, and autophagy. So what I'm going to describe to you is two main topics. One of them showing a methodology that we have developed, the Pca methodology, which provides a dynamic view of protein-to-protein interaction in the PCD map, and we can follow the PPI, the protein-protein interaction, in real time in cells, looking on the entire 100 proteins. This led us to a few achievements. First of all, a global view of all the interaction, including interaction between apoptosis and autophagy. Then we could follow the PPIs in response to stress responses. And it also led us to novel pathway discovery There are a few. I decided to talk on one of the DAP kinases as a novel discovery linking stress response to autophagy, to the basic machinery of autophagy. So this would be the first part. And then I will tell you my second part, how we harness this huge information that we have on the entire map, how we harness it in order to vision some therapeutic strategies in precision cancer therapy. Can we do that? Can we use this information? So the methodology that I want to tell you was developed by Yuval Gilad, a student, a very talented student, ex-student in the lab. And it is called PCA, for protein fragment complementation assay. So it's a simple split luciferase assay that you are familiar with. But we developed specific constructs which are based on humanized luciferase enzyme, which are very sensitive, highly specific, with a minimal noise to signal, a minimal noise in the system. And what we do here, we take the 100 proteins that participate in programmed cell death. We link to each protein a fragment of luciferase, either at the C terminus or at the N terminus. If protein A and protein B interact, then we will get luminescence. And we can assess at real time the luminescent signal that we get whenever we introduce the entire battery of constructs into the cells. So first of all, we wanted to be sure that indeed it works and that we can follow the dynamics of interaction. So we took a well-known example, ICAD-CAD, two proteins that at basal conditions, they are there in cells as a complex. So it's a complex of protein. Now, indeed, if we look on the luminescence signal using those reporters, we see nice luminescence. So yes, it lights up. Now, if we expose the cell to stausporine, for instance, we activate caspases, the ICAD is degraded, and you can see that it is degraded, either the reporter or the endogenous genes, and indeed, we lose the signal. So you can see that the signal is now lost. And if we put an inhibitor of caspases and we prevent the cleavage, we get back the signal. And this is between 1 to 4 hours after adding the stress. So this is one pair that convinces us that we can follow the dynamics of the complex formation, or this PPI formation, in this system. So Yuval prepared the library, the Pca library. So we can see here the apoptotic genes, the extrinsic, intrinsic, the caspases, the BCL2 family, and so on. You can see the autophagic genes that I mentioned here and there, the Becklin, the ULK, and so on. And we also introduced the DAP genes because we are working for many years on DAP genes, and we think that there are, among their function, there are some of the second layer of regulation that couples the stress signal to the basic machinery. And what Yuval found, what he did, he took all those proteins and he puts in each well a couple, one couple. So if we have, let's say, 65 proteins in this type 1 library, so we have all possible combinations are tested, so we have 3,600 combinations to test. It's not a big deal. You can do it by a robot, or you can do it even at the beginning we did it without a robot. So what we do, we put here the different pair combinations of all the library. On top of that, we play the cells. After 24 hours, we read the luminescence, and now we know which protein interacts with which protein because it's random. And this is how we get this heat map. So you can see from 3,600 combinations, we could get 131 hits, which means that 121 positive hits are lighting up. Those are the proteins that interact with each other. Out of what was known at that time, we had 85 out of what was known at that time. But the important thing was that we discovered 46 new interactions. And this was for us very, very important because now we can get into those 46 interactions and add more errors and understand more things in the system. So here you see the network view of what I showed you before, those 131 interactions. Here are the apoptotic proteins, here are the autophagic genes, and here are the DAP genes. All the red arrows indicate the new interactions, and the dark arrows, the known. So you can see that, for instance, in the BCL2 family, there was a lot of knowledge, and we didn't discover any new interaction. The same is from the ubiquitin-like activity that elongate the membrane in the autophagosome, no interactions. But yes, we got a lot of interaction, new interactions, which between, let's say, autophagy, apoptosis and autophagy. You can see all those red, and between the DAP genes and the basic machinery of autophagy. So we were happy that we have succeeded to reveal new things. So this strategy opened three main directions, and I will describe them very briefly, what we got from here. First of all, we got a global view, and novel PPIs, novel protein-to-protein interaction, within apoptosis, within autophagy, and very important, between them. For instance, the green ones are between autophagy and the apoptosis, and we discovered by that means 14 points of interface between apoptosis and autophagy, which adds a lot of information on what was done and known before. So this was one achievement. The second achievement was, okay, let's see if we can monitor dynamics, but not just looking on one pair, but looking on many, many proteins. Can we monitor dynamics? So what we do, we take the Pca library, and we now either assess it under basal conditions or in response to a trigger. For instance, starving the cells for amino acid. We know that autophagy is turned on. We want to know what happens to autophagy, to apoptosis under these conditions. Can we see a dynamic view now of many proteins simultaneously? So I brought you one example, which is a very important example for us, is the ULK1, a serine-threonine kinase that participates in the nucleation process of autophagy. There was a lot of information about how it's being activated by mTOR inhibition and so on. Little information about which are the substrates of this important nuclease. So, kinase, sorry. So this is a plate map where we tested ULK1 with each of those proteins. And we got a nice profile showing that if we amino acid starve the cells for 2 hours, turn on autophagy, we can see new interactions coming up with those genes. And for instance, from here, we focused on the WIP2, and we discovered some new interaction between ULK1 and WIP2 during autophagy. I won't tell you the details, but just showing you that this platform provides a quantitative reporters that can follow changes in PPI in real time in cells at the global level in response to stress, which for us was second milestone using this technology. The third milestone that I will focus and zoom into it is discovering new interactions that were not known. And what we have done so far extensively, discovering what is the importance of DAP kinase 2, a serine-threonine kinase, interacting now with ATG14, a protein that sits in the nucleation phase of autophagy. What is the relevance of that? What is the importance of this interaction? So let me go through it step by step. So what is ATG14? ATG14 is part of the complex, of a Becklin-1 complex. This is the complex which is very important in the nucleation process. It turns on PI3 kinase. It's a lipid kinase that produces this phosphatidylinosyl 3-phosphate. Are those yellow spots here. And this phosphorylation, this lipid phosphorylation is very important in docking the subsequent proteins, those proteins that participate in the elongation. So it's a very important step from the initiation to the elongation and the closure of the autophagosome. So once we realized that DAP kinase 2 is interacting with ATG14, we wanted to know what is the implication of this phenomena. So first of all, what is DAP kinase 2? It's a member of the DAP kinase family that we have discovered many years ago. And it's a calmodulin, calcium calmodulin dependent kinase. So it needs calcium and calmodulin in order to be activated. And here you can see the 3D structure. So this is the calmodulin that blocks the catalytic cleft as long as there is no calmodulin binding domain that blocks the catalytic cleft, as long as there is no calcium in the system. Whenever calcium calmodulin come, they bind to this region and pulls away the calmodulin regulatory domain from the catalytic cleft. This is the upper lobe, the lower lobe, the calmodulin regulatory domain, and this is the silent form of the kinase. So in addition, there is a phosphorylation, a negative autophosphorylation in the calmodulin regulatory domain at serine 308. And this phosphorylation, autophosphorylation, locks the calmodulin binding domain in the catalytic cleft. So that double mechanism to lock the enzyme and to prevent its activation under basal conditions. And we knew that it is somehow connected to autophagy because previously we saw that either overexpression or knockdown can do something to autophagy. So the big question was, what is the relevance of the interaction between DAP kinase and the ATG14 that we have discovered by this Pca methodology? So I will go to it very quickly. We have recently published that. So what we saw, first of all, that indeed DAP kinase 2 interact with ATG14. We can see it in those co-immunoprecipitation assay. They do bind. It's not just the reporter genes. They bind in the cells. We saw, and we looked, who is phosphorylated by DAP kinase. This is a kinase. It phosphorylates a substrate. Who is the substrate? At the beginning, we were sure that ATG14 will be the substrate because ATG14 is the guy that binds to DAP kinase 2. But we discovered that DAP kinase phosphorylates Beclin-1, the scaffold protein that is very important to activate the PI3 kinase. It phosphorylates at a certain position that we have elaborated antibodies to that position, threonine 119 in Beclin. And this is very important because this is a critical position in Beclin. This is a critical position that once you hit it, you inactivate the inactive, let's explain it better. This complex is inhibited by BCL-XL, by members of the BCL2 family. This is another example how we get a apoptotic protein interfering with the autophagic machinery. So others have shown that BCL-X inhibits Beclin-1, and it does so by interacting with the BH3 domain in Beclin. Beclin has a BH3 domain, binding domain, like BAX, BAC, and the other apoptotic proteins. And this binds, and by that means blocks the activity of Beclin. Now, we have shown that DAP kinase 2 phosphorylates Beclin in this threonine. This threonine is very critical to inhibit this binding. And by that means, we release the complex from its inhibitory element. So we showed it by several strategies. For instance, here we were using a phosphomimicry mutant, and the reporter, the PCA reporters, showing that indeed we lose the interaction with Beclin, with BCL2 if we phosphorylate Beclin at this particular position. Again, it's a long story. I'm shortening it here. So at that stage, we knew that DAP kinase indeed interacts with ATG14. It phosphorylates Beclin in this important position, and this phosphorylation relieves the BCL2 and activates the system, leading to autophagy. And the big question was, okay, so how this axis is activated during autophagy? Because I told you one of our goals is to understand the coupling between the stress signal and the basic machinery of autophagy. So how does? So again, it's a very long story. It was done by Uchi Lo, a PhD student in the group, and we have published that this year. And again, to cut a long story short, I would just tell you that Ruti discovered that AMP kinase is a central metabolic sensor, which is a central metabolic sensor, is the kinase that phosphorylates DAP kinase 2, and by that means, it activates DAP kinase 2. So we are dealing here with transphosphorylation, a metabolic sensor that is activated during metabolic stress, is phosphorylating DAP kinase, and it's phosphorylating DAP kinase again at a very important position. And I will tell you very briefly about this position. It's a position 289, which is very critical in the structure of the protein. And you can see here that the kinase domain, now I'm showing you the structure of the kinase domain, it has this calmodulin binding domain, and it has small short loop where we have identified the phosphorylation of DAP kinase 2 by AMP kinase. So during stress, AMP kinase phosphorylates here. And this, what we have shown, that is an important hinge that discriminates between the calmodulin regulatory domain and the backbone of the kinase. So we assume that this phosphorylation will activate the catalytic activity of the kinase very critically. And indeed, this was the case. We could see that indeed, if we assess the catalytic activity of DAP kinase 2, we see that it is very, very strongly activated by this specific phosphorylation. And more importantly, we have shown that the AMP DAP kinase axis promotes the backlink phosphorylation at position 119, which is so important. And you can see it here and quantifying here. So again, to make a long story short, we have shown that this phosphorylation reduced the homodimerization of DAP kinase 2. This is in the silent form, it's an homodimer. It abolished the inhibitory serine 308 phosphorylation, and it increases the catalytic domain, but the important thing, in a calcium calmodulin independent manner. We don't need a calmodulin here. We have this phosphorylation that opens up the calmodulin regulatory domain without needing the calmodulin. And we have proposed a model where we have two ways to activate DAP kinase 2. The canonical way, where the calmodulin binds to the calmodulin regulatory domain and releases it from the catalytic lift. So this is the canonical way. And now we found by this strategy that there is a phosphorylation, a second transphosphorylation. The 308 is an autophosphorylation. This is transphosphorylation that opens up the calmodulin regulatory domain independent of calcium calmodulin. And it happens in response to a metabolic stress when AMP kinase is activated in the system. So to summarize that, so what by identifying a new interaction between ATG14 and DAP kinase 2, we came up with a pathway, metabolic stress, turning on AMP kinase phosphorylating DAP kinase 2 at a very critical phase that now opens the catalytic left without calcium calmodulin, and then DAP kinase 2 phosphorylates backlink again in a critical phase and releases the inhibitory effect of BCL-XL and induces autophagy. So to summarize this part, how much time I have? 15? 15, good. Okay, so to summarize this part, I've shown you that this Pca library, the protein fragment complementation library, enables rapid and accurate detection of protein-to-protein interaction within and between the apoptotic and autophagic model, which was the first thing that we were very happy about it. It's a dynamic system, so we can follow the changes in the profile of all the tested interaction simultaneously in response to certain steps. So this is very important when we now want to convert the static map that people have described from hundreds of publications to something dynamic in a certain step, in a certain cell, in response to a certain signal. We identify in a few interactions. I've described to you this interaction, the 14 DAP kinase 2, which led to the discovery of a pathway that links metabolic stress to autophagic induction. And we were surprised to see that we have 14 novel direct interactions between apoptotic and autophagic proteins, which means that the connectivity between apoptosis and autophagy is stronger than initially assumed. And we think that this connectivity is important whenever a cell has to decide whether to live or die in response to